So, hello everybody. My name is Sarah Butcher and I'm from the University of Helsinki in Finland. And I'm very happy to be hosting today this webinar on behalf of the Instruct-ERIC Centre in Finland, which covers Helsinki, Oulu and the University of Eastern Finland. This is the 9th webinar in the series of Structure Meets Function. I'll start with a short introduction into Instruct and then into the Instruct-ERIC Centre in Finland. For those of you who are not aware, the Instruct is a European research infrastructure consortium which is focusing on structural biology. It operates a number of different programs which are illustrated here by these 4 emojis. The most important of those is access to high-end structural biology research facilities throughout our network in a number of different centres. In addition, it provides training courses, workshops, and these are focused especially in integrated structural biology, so bringing together different types of methods and approaches to address more complex issues. In addition to this short-term access, you can also apply to a facility to undergo an internship, and these are 3 to 6 months stays in one of these different facilities where you can really get it not only into how the machines are working, but also into addressing a complex project which would need a lot of input from the staff scientists which are present. In addition, we do have a research and development program, and the idea of this is that, say you've got an idea for a new technology which you think will be a real breakthrough in structural biology. So maybe you need a bit of seed funding to help you develop this new technology. And so we're very interested in supporting these sorts of efforts as well. And that's the R&D funding program. Now, in addition to these 4 activities, you can see that from this map of the different member states, which are part of this ERIC, you can see that a lot of coordination needs to go on, and this coordination activity is a very important part of the ERIC. It's coordination between the different centres, the different countries taking part, international organisations such as the EMBL, which is also a member in the ERIC, and also as a way to influence the decision makers, policy makers in the EU, in the national governments, and also interacting with things like the European Open Science Cloud, which are tremendously important for all of us in terms of open access to science. In terms of the members, so you can see that we're distributed all over Europe. And up here, there's this huge country, which is absolutely to size, which is Finland. We're very, very small in terms of numbers, but we're very large in terms of landmass. And as you see, we're also one of the most easterly centres in Instruct, along with Israel down here. So within each country, you can see there are these red dots. These are the 11 centres which offer the access to facilities. And so you can travel to any of these. Now, of course, during the past year, most of the access has been remote. Some of the centres have been open or closed for visits, depending on what is the status of the pandemic in those centres at the time. But we all offer some sort of remote access or send-in facility. Right, so if we then move on to the Instruct Centre in Finland, so this is a so-called distributed centre, which means that physically we're located in 3 different positions, up near Lapland in Oulu, close to the Russian border in Joensuu, and then down in the south in Helsinki. In the University of Eastern Finland, The main offering is from native mass spectrometry. University of Helsinki, we have a broader range of offerings in biomolecular complex purification, cryo-EM, cryo-correlative and light and electron microscopy, NMR, segmental labeling, single-cell proteomics, biomass spectrometry, and biophysical characterization. And then in the University of Oulu, molecular biophysics, macromolecular crystallization, X-ray diffraction, and especially data management for X-ray crystallography. I especially want to highlight things which are unique within the ERIC as far as we are aware. So it's especially the segmental labeling, which is going on in Helsinki, the single-cell proteomics by mass spectrometry, and the biomolecular complex purification. Today, we will have 3 speakers, one from each of these 3 different sites, just to showcase some of the science that you can do by utilizing these different technologies. And I hope this way you'll get a really practical understanding of what is possible. First, before introducing the speakers, however, I need to introduce the 10th webinar in the series, and this will be conducted by the Instruct Centre in France. There are 2 centres in France, and this is the first one. And this seminar will be on the 12th of May, 2021, at the same time as the current seminar. And remember, all of these seminars are also recorded and available from the ARIA website, which is the main site for the Instruct-ERIC. So today, we are going to kick off with Minna Poranen. So she's going to tell us about the facility that she's running called Biocomplex, and the sort of examples of sample preparation strategies for biomolecular complexes, so large objects with many, many thousands of components. After that, we're going to run on to Lari Lehtiö in Oulu, who's going to talk about some of his work on crystallography. And then Yuhar Rovenan will end with a tour de force on applications of high-resolution native mass spectrometry. So without further ado, I want to remind you, please, will you use the Q&A session if you have questions? And we'll have a question and answer session after each talk, and I will then decide which questions are then asked to the speakers. All right, so I will stop sharing. Minna, please take over. Thank you.
So thank you, Sarah, and good morning, everyone. So in the Biocomplex facility of the University of Helsinki, we develop methods and provide services for the purification and separation of large macromolecular assemblies, such as ribonucleoprotein complexes, lipid protein vesicles, and viruses. And the key technologies that we have are preparative ultracentrifugation, monolithic chromatography, and asymmetrical flow field flow fractionation, or AF4. And these techniques can be used separately or in different combinations to produce high quality samples from fragile assemblies of macromolecules for structural and functional analysis, but also for other purposes, like for example, for biomedical research. And the samples produced in our facility can be also directly placed on cryo-EM grids in the cryo-EM facility led by Sarah Butcher that is located in the very same building. Asymmetrical flow field flow fractionation is a new technology in the Instruct-ERIC service catalog. This is a very gentle method for the separation of biomolecules. The separation is done in a liquid phase, and the mobile phase composition can be adjusted so that it best supports the integrity of the studied molecules or complexes. The sample, which can be, for example, a lysate of cell, is injected into the AF4 channel, and the mobility of the components is controlled with using different flows, and the diffusion coefficient and the size of the studied particles then affects the mobility in the channel. There is a large dynamic size range for separation, so for small molecules up to particles that are 1 micrometer in diameter. And we have just recently obtained a very new instrument in our facility where there is an option for online data collection using many different type of detectors attached to the instrument. And here are shown just some of our recent analysis of a viral particle showing examples of the collected data on the particle conformation, molecular weight, concentration of particles in the peak fraction, and heterogeneity of the sample studied. And this setup is available both in analytical and semi-preparative mode. One of the viruses that we are studied in our laboratory is an enveloped double-stranded RNA bacteriophage 5/6. This virus shares a similar inner capsid architecture and intracapsid genome replication and transcription strategy with the eukaryotic double-stranded RNA viruses like rotavirus, Pluton virus, and orthoreovirus. The inner capsid, which we also call the polymerase complex, is enclosed into a second protein shell, the nucleocapsid surface cell, to form the nucleocapsid particle, which is then enclosed into protein lipid envelope in the infectious virion. The polymerase subunits, the RNA-dependent RNA polymerase, is the key component of the polymerase complex, and it has been extensively studied both using biochemical and structural methods during the past years. And now we are interested to understand how these polymerase subunits are, first of all, incorporated into the polymerase complex particles and how they operate during the life cycle of the virus as a part of these particles. For this purpose, we use an vitro self-assembly system where we start from purified protein components produced using recombinant expression system. When these are mixed in the presence of polyethylenyl glycol, they self-assemble into empty polymerase complex particles. And these particles can then be purified using regional centrifugation. And the purified particles are used for structural and functional analysis. In the presence of ATP, these particles can package the positive strand genomic precursor molecules of the viral genome. If all the nucleotides are provided, these packed RNA molecules are replicated into double-stranded form within the particle by the polymerase subunit. And then if later on, this double-stranded RNA can be used as a template for the transcription reaction. And during this process, the particle undergoes major structural conformational changes, and its internal volume is substantially increased. Using the self-assembly system, it's possible to manipulate the composition of the polymerase complexes. And Chowi San, a postdoctoral fellow from my group, make this kind of self-assembly reactions where she applied increasing concentration of the polymerase subunits in the reaction mixture. And using this method, she was able to produce particles that contain increasing number of the polymerase subunits enclosed into these particles. So the normal copy number for the polymerases in the 5/6 virions and these kind of polymerase complex particles is 10. But up to 19 copies of the polymerase subunit could be incorporated in these particles using the self-assembly system. And the activity of these particles was tested using the packaging and replication analysis. And here is shown the results from the particles that contain approximately 10 copies of the polymerase subunit, so they can replicate the RNA to produce the dsRNA molecules and also produce transcripts of the 2 RNA segments. And similar activities were observed with particles containing excess of the polymerase subunits. However, if there was substantially low amount of the polymerases in these particles, then this was affected to the transcription activity of these complexes. The In vitro self-assembled particles were then also studied using cryo-electron microscopy, and here the key person was Serban Ilka, a PhD student of the University of Oxford, supervised by Juha Huiskonen and David Stewart. And in this cryo-electron microscopic reconstruction, Serban was able to observe density for the polymerase subunit within these particles. And using localized reconstruction method, he was able to define the precise position for the polymerases in these particles. They are located at the true-false symmetry axis, making specific contacts with neighboring five-fold vertices that are orientated inwards in these empty polymerase complex particles. And when these kind of particles were analyzed using regional centrifugation, we observe a single light scattering band. However, if we analyze particles containing a reduced number of the polymerase subunits or no polymerase at all, we see an extra band. And this band corresponds to these kind of slightly expanded particles which have slower sedimentation velocity. So this data suggests that indeed these polymerase subunits, by bridging together these five-fold vertices, can stabilize the conformation of these empty capsids. Serban was also able to identify potential binding sites and residues involved in the binding between the polymerase subunit and the major capsid protein on both sides of the polymerase protein. And these proteins or residues in these polymerases were mutated using site-directed mutagenesis. And then these were analyzed using the self-assembly system. And we see that if these surface mutant forms of the polymerase were studied, they were incorporated in less efficiency than the wild-type polymerase subunit into these particles. So it suggests that the predicted polymerase major capsid protein interaction sites are also essential for the polymerase complex incorporation into these particles during capsid assembly. Alternative way to obtain viral subparticles is the controlled disassembly of purified viruses, and this can be achieved by different chemical treatments. Here we have isolated the 5,6 nucleocapsids, the double-layered particles using Triton extraction and purification using monolithic chromatography. And Serban studied these particles again using cryo-electron microscopy, and he was able to build an atomic model for 90% of the total 5,6 double-stranded RNA genome. The genome is organized into 5 layers around the three-fold symmetry axis, as shown here, the layers. However, what was surprising for us that we were unable to see any density for the polymerase in these structures, which suggests that the polymerase is displaced from its original site during the genome replication or genome packaging process. So there must be changes in the protein-protein interactions during the different functional stages of this complex. In addition to these structural and functional studies, we are also interested in the structural conservation in viral proteins and especially in the viral RNA-dependent RNA polymerases. So currently there are polymerase structures available from 42 different RNA virus species representing 16 viral families. These are listed here, so including double-stranded RNA viruses, positive-strand RNA viruses, and negative-strand RNA viruses. And RNA virus genomes evolve fast, which means that there is no detectable amino acid sequence conservation among distantly related viral RNA-dependent RNA polymerase genes, and therefore sequence-based comparison methods can be demanding and can be supported by structure-based analysis. And for the structural analysis or structural comparison of these polymerases, we use a program called Homologous Structural Finder, which has been developed by Janne Ramantti from our university and David Stewart from Oxford University. And with this program, we were able to identify a common structural core of 231 residues that is conserved across all these viral polymerase structures, and it's depicted here on the poliovirus polymerase. The conserved or equivalent residues in the thumb, palm, and fingers domain are colored in this structure. And this covers over 50% of the poliovirus polymerase structure and some 25%, for example, from the SARS coronavirus 2 polymerase structure. So there is substantial structural conservation in these polymerases. Using this information, we also make a structure-based phylogenetic tree for viral RNA-dependent RNA polymerase, which is shown here. And we see that the polymerase structures are clustered into two main groups. And among the first group are polymerases that mainly use conservative transcription mechanisms and have 4-tunneled polymerase structures, whereas in the group cluster 2, we have polymerases that use semi-conservative transcription mechanisms and have 3-tunneled polymerase structure. We also see clustering based on the priming mechanism. So here are polymerases that use protein primer, those that use de novo initiation, using self-priming, or then in some cases, the negative strand RNA viruses can also use an RNA primer. So as a conclusion, high quality samples of macromolecular complexes for structural and functional analysis can be obtained using ultracentrifugation monolithic chromatography or AF4. And structural and functional studies on viral polymerase complex have provided new information on key interactions within these particles. And we have also observed changes in such interactions, which underlines the importance of studying different functional stages of biological complexes to fully understand the molecular mechanisms. And structure-based phylogenetic analysis was used to clarify long-distance relationships between viral RNA-dependent RNA polymerases, and the data shows strong link between structural evolution and functional specialization in these proteins. And I would like to thank all the people who have involved in my laboratory and especially in the Biocomplex facility, Katra Eskelinen, who is our expert in the AF4 technology, Janne Ravantti-Heli-Mönttinen, who have been involved in the structure-based comparison study, and then Serban Ilka and all the other people in Oxford University who have been doing the cryo-EM analysis that I presented. So thank you.
Thank you very much, Minna. That was a very nice, clear talk. So we have one question in the Q&A. Are the reconstituted particles infectious?
So in these reconstituted particles, we can add also a second protein layer, the nucleocapsid surface cell protein layer. And actually these nucleocapsid particles are infectious to host cells spheroplasts. So the normal host is a gram-negative bacteria, which have this outer and inner membrane. But if we remove the outer membrane, then the nucleocapsid can infect these spheroplast cells. So they are infectious, and their activity can be studied using plaque assay then.
Yeah, so that's obviously a huge advantage of this particular system. Mm-hmm. So if you had to, Minna, maybe in a nutshell, say, why should we use AF4? Especially the new instrument. What's your selling point?
Well, I think that if you are considering some very fragile complex that is easily dissociated, I think this would be the best method, because really there is no interaction with any airy surfaces, and you can select the buffer conditions so that they best support the stability of the particle, and they can be maintained throughout the analysis or the separation, the conditions. So I think it's, in that sense, it's very good. And also the data collection now, that you can monitor the quality of your sample and the heterogeneity, aggregation, and these kind of factors. So.
And in terms of the scale, is this then enough to actually produce something for, say, crystallography down the line? I think that's related. Bruno has also said for the AF4, what's the volume and the amount needed? So if you could answer that multi-part question, please.
I think that, of course, our focus is more like on the larger complexes. So maybe the cryo-EM would be the next step of analysis, not necessarily crystallography. But the sample is a bit diluted, gets a bit diluted there. But of course, it can be concentrated afterwards. But yes, it can be used for preparation of samples for cryo-EM, definitely.
And there's another question now that have you purified other complexes like proteins involved in biosynthetic machinery or only viral particles?
Yes, we have purified, and of course our users are purifying different type of samples like ribosomes and extracellular vesicles, and different type of complexes are purified using these services.
And also, Bruno would like to know how much can you actually recover from the...
This is a bit specific question for me, but I think that the recovery from the AF4 is high. From the monolithic chromatography, we usually lose quite a lot, but it depends, of course, on the material also.
Yeah. So the previous generation of the AF4 has been primarily an analytical machine, but we'd like to really emphasize now that this new machine is really of a scale for preparative as well.
Yeah, so we have different channels for preparative and analytical purposes.
Then I'd like to thank you once again, Minna, and hand the floor over to Lari, please. Thank you.
Thank you, and good afternoon, everybody. I'm going to talk about ADP-ribosylation and how this post-translational modification is switched on at the DNA damage site. This has been studied in our facility and in my team, and the Instruct facility in Oulu is hosted by the Biocenter Oulu Structural Biology core facility, and we provide services, as Sarah mentioned in the beginning, in molecular biophysics jointly with the protein analysis core facility, crystallization, X-ray diffraction connected to that, and then to crystallographic data management. And this crystallographic data management I would like to especially highlight here. We are developing a software called IceBear. It's a research data management for structural biology. It's a data management system containing everything from basically the protein construct to the deposition, publication to raw data, and so forth. It interacts with the imagers, so we have imported crystallization imagers. We can annotate them, but it also allows easy communication between team members. It works offline, so our users actually who are using the facility now at the moment remotely doing crystallization, they also have access to this software from their own home or laboratory. It works with several automated imagers, and it has built-in pin-pack dual sample management system, which allows us to ship samples virtually to synchrotrons. So the samples travel by plane. We don't travel at the moment, but the metadata regarding those samples is transferred directly to the database at the synchrotron. So there's no basically typing at all at this stage. This was recently published earlier this year, the publication regarding this Instruct Ultra-funded project to establish the communication. And there's web pages available where you can go and you can actually test the system with as a demo account. And how it connects to this rest of the presentation is that in the laboratory, what you do when you are preparing a sample for the shipment, you actually read the barcodes of the pins, of the dewars and the pucks for the crystals, and then you can pick up the crystal, and with one sweep it goes to when you have picked up the crystal, it goes to the pin, to the puck, and then automatically to the dewar. Of course, you can modify this however you feel like afterwards. And then the sample is shipped to the synchrotron facility by courier typically, and we prepare the sample shipment also in silico using the connection to IceBiop. And for instance, here you have seen that there's a demo crystal proteinase K, and it has been actually the same crystal has been picked up and shipped 3 different times to Diamond Light Source when we were testing this system. And you can actually go and click this link, log into Diamond, and you see the data collection results at the Diamond Light Source. So yesterday evening, I checked the structure that I'm going to talk about next, and actually with the PDP deposition codes, which is here, I can see that it's available in the different PDP sites. Of course, now currently this is on hold, but and there's a link to Proteopedia, and it's actually collected from 4 different crystals, collected all at the Diamond Light Source, and you can go back and look at what was actually happening in that particular data collection session when you collected the data. It also shows you naturally the crystallization conditions and also data sets, constructs, and protein acronyms and whatnot, and you can add notes here. So this structure that I'm going to talk about is from 4 different crystals actually. And this is basically the structure. So it's this DNA binding enzyme. So it recognizes the DNA damage. One domain is recognizing the DNA damage. There's actually a break here. Then you have an autoinhibitory regulatory domain, and then you have the catalytic domain, which binds substrate NAD. And this NAD is then used to post-translationally modify itself and other proteins. And what happens here is actually that it generates this large polymer of ADP-ribose. It can be linear, branched, it can be only one single unit of ADP-ribose attached to a target protein, and this depends on the enzyme that is carrying out the reaction. We talk about the PARPs in general, and usually we refer to the DNA damage dependent PARPs, but there actually is 17 members in the family, and they have different type of activity. Typically, they modify acidic residues like aspartate or glutamate, but some of them actually modify also serine, and this is actually critical in DNA repair, because during the DNA damage recognition, the modification changes from acidic residues to a serine, which I will show you later. There's also other proteins involved that recognize this signal, because you generate at the DNA damage site massive modification, then you actually have these other protein domains are recruited to the site of the damage by these other domains. There's macro domains, WWE domains, and so forth. Human PARPs, they have rather conserved catalytic domain, which is usually at the C terminus. So this is related to diphtheria toxin. That's why it's sometimes called ARTD. One of them have it here. But the DNA damage dependent PARPs are actually these first ones. They have the same structure. They have the catalytic domain, then a regulatory domain that we think is actually an autoinhibitory domain, then have a VGR domain that contributes to the DNA binding, and then the N-terminus is different. In part 1, you have zinc fingers recognizing the DNA damage site, but in these other 2, you have a disordered N-terminus, which has a high affinity to DNA, but it's disordered, so it doesn't have a structure. There's some motifs recognized here, but nevertheless. So the catalytic fragment, actually, it is you have a NAD binding site here, and then you have the acceptor protein or polymer binding here, which is then elongated. And most of the enzymes, they have only this domain and then some additional domains. But all these 4, especially the 3 first ones, they have the regulatory domain here, which is the alpha helical domain next to the catalytic site. The NAD is actually binding here. So this is, we think, is an inhibitory. Despite that we have had 20 years these structures available, there's no structure of an actual NAD binding to this sort of a structure that I'm showing here. I will focus next to the regulatory domain and what is happening actually when this enzyme is binding to the DNA. So the previous studies have all reported this inactive, what we think is an inactive conformation of the enzyme. The first structure of a PARP1 was published in 2012 in Science, where you actually have most of these domains. These white ones are not present, but here we have the HD, or the regulatory domain, and catalytic domain, and VGR domain. And we use this structure to model our inactive conformation of the PARP2 enzyme. There's a structure of the substrate analog, but this is only present when you remove completely the helical domain, because there's some clash here. So something has to happen when the DNA activates the enzyme. There's also another protein, present, which is a histone pyrrolation factor. And this actually forms a joint active site together with these enzymes. And this is the key to change the activity from the acidic residue to a serine residue. And this was also this structure published last year, but it's also, again, when you don't have the regulatory domain at all here, then the HPF1 can bind. And we have shown previously that actually specifically DNA damage so that it bridges both sides of the damage, and it's actually activated by the 5' phosphate. When you have the 5' phosphate binding to this tyrosine, then you have a boost in the activity. So we were able to solve the structure of a PARP2 complex in complex with this activating DNA. And this was actually from 4 crystals, 26 data sets, because we discovered that the crystals are diffracting very not uniformly. But we managed to at diamond collect using microfocus beam quite a decent data set to 2.8 angstrom resolution. And actually, when you look at the biological unit, then you have actually two VGR domains binding to this DNA damage site and 2 copies of PARP2. So they form this kind of a joint, joining the blunt ends of the DNA. And basically, this is a zoom-in picture of this one showing that the 5' phosphate is binding to these key residues at the VGR domain. How is this then boosting the activity of this domain far away? That we don't exactly know yet. However, we were able to capture this because there was actually another DNA end TA pair at the end of the DNA was binding to this catalytic domain, and it's actually binding to the active site, keeping this catalytic domain in place. And we have actually shown that thymidine, they inhibit this enzyme, so they mimic the substrate in a way. This is relevant because it has been shown also that PARP2 can modify DNA, but it modifies the same DNA where it's binding to. So if you imagine that the VGR domain is binding here, then this domain is able to modify this end of the DNA, implying that there's a lot of mobility in this domain. So we use the PARP1 structure, and all these structures that are of PARP enzymes that are in the BDP, they are basically mimicking this inactive structure that we think is kind of the basic unit when the enzyme recognizes DNA, but then activation happens. And this we think that we saw in our structure. So there's a big movement in this regulatory inhibitory domain. And this is again a zoom-in view. When you look at the inactive model, then basically this NAD cannot bind to this active site at all because there's this helix blocking the access, and there's actually clash between some residues and this helix. In our structure, this catalytic domain is completely free to bind the end substrate NAD and catalyze the reaction. And actually, the conformations that we see, conformational changes, are quite big in the structures when you compare them. We also did some mutagenesis to kind of probe this, and we pinpointed one residue, this glutamate here at the regulatory domain. It's interacting with the backbone of the catalytic domain, and when we modify this and change this to an alanine, then there's a bit of a boost in the activity, but not that much. But when we change it to positively charged arginine, then actually there's a repulsion between these domains, and we have a hyperactive enzyme even in the presence, even without DNA. So it makes the enzyme completely active, even though there's no activation needed, and this is quite far away from the DNA binding site. We also tested this in gel, and here you can see that there is basically no activity here, but when we have the mutant, even without DNA, then we have the polymers forming. So our view and putting this structure to what we know about these enzymes in during the last 20 years and their structural studies, then we think that the first thing is happening, that these enzymes are scanning the DNA, they find the break and recognize the DNA damage. And there's structures available of these DNA protein complexes with different domains and so on. And then activation happens. So based on there's also some other data showing that there's something happening in the enzyme. And in our crystal structure, we see that there's actually big movement happening here. So first of all, we have the inactive structure, and this change in the location of this domain is quite massive. We think that we have only captured this one conformation, and this domain is likely rather rather flexible and can move around and recognize substrate proteins and even the DNA that this is binding to. Then the NAD is binding. Then the HbF1 is binding, forming the joint active site, substrate protein, which is mimicked by the DNA. And then you have the elongation of the polymer to make this up to 200 ADP-ribose unit long polymer. And when you create that one, of course, the HbF1 has to dissociate from the protein. So we have the joint active site, binding of the NAD, joint active site with HbF1. And then we have the substrate protein binding. There's actually some data showing that there's some melting of helices of HbF1 and PARP2 and allowing this sort of a macromolecule to bind here. And then you have the PAR elongation. This was actually one of the first structures where they showed that then the acceptor site, there's actually NAD binding here. With that, I want to thank everybody, especially regarding PARP2, our team, Eseogo, Mirko, Albert, who has been involved in this PARP2 studies. And regarding IceBear, of course, we are collaborating with a lot of facilities within Instruct and outside, and putting IceBear has also been installed in multiple places already. And we are developing so-called Icebox, which is a cloud implementation, allowing even small labs to easily start to use such a software in data management. With that, thank you, and I would like to answer any questions you may have.
Okay, great. Thanks very much, Lari, for a really clear talk, and I hope the structure is going to be published very soon.
I hope
Any of the reviewers who are listening. So there is a question in the Q&A which says, PARP interaction with DNA more on open end or the central part of the DNA? In other words, is it nick or break specific?
Well, it depends a bit. I mean, what we see here is the joining of two plant ends together. And we got this complex because we have the 5' phosphate only in one of the ends. So here is phosphate, here is no phosphate. There is the TA pair, and that's why it's packing in the crystal. We have actually gotten crystals with multiple lengths of DNA and a lot of iteration to get some diffraction. But in PARP2, it seems that it's binding to this end and joining DNA ends together. But we don't know if this happens in cells. So our interpretation is that this is mimicking a nick because there's a break here. We don't know if this joining of the DNA ends happens, but it's possible.
Have you thought about any ways or approaches in which you could test that?
Yes, we have, and I'm sure other people have also, but nothing has come out so far.
Okay, so Bruno, you have a hand up. Please go ahead.
Very interesting structure. I had also a similar question about the interaction. You mentioned that it may also recognize modified DNA. So how would that happen from the side then, or and what would the implication be? So
it can recognize some defects in the DNA, but this is most of the data is based on in vitro data showing that the enzyme can be activated by certain forms of the DNA. We know that there's some basic activity of the enzyme. We have activity without the DNA. There's some activity. We see activity when you don't have the 5' phosphate, but it's just massively boosted when you have the 5' phosphate in this particular enzyme. In contrast, PARP1, it doesn't require this phosphate. So it recognizes via different mechanism. It has zinc fingers and additional domains there. And I think also that this other DNA that we have in this structure is actually on the place of the PARP1 zinc fingers in the structure, where it is actually recognizing the end of the DNA.
Okay.
And with PARP3, we don't have that much data yet how this actually recognizes the DNA, but likely very similar to PARP2.
Okay, thanks very much, Larry. So there's a couple of questions now coming up on IceBear. So if we take the first one, do you need to upload the structure information to IceBear, or will it be updated automatically from other databases?
So naturally, when you are carrying out crystallization, you put the construct information. We have the philosophy that that we don't require anything, but we just notify the user that please upload if you want. So that's not required, but usually people put the construct information because then you have it in place when you have the data. Then shipment of the crystal is automatically, it knows where you have the what kind of a crystallization conditions you have used. We can import most of this data automatically. Imagers, depending on the imager, then we either copy the data, usually from our imagers, we copy the images over, and shipments to IcePyB, then we automatically have links from the crystal shipment to the IcePyB pages, and at least max 4 they have now links back to IceBear also. So when you are collecting data at the beamline, you can go and check the crystallization conditions or the actual crystal or whatever. For the raw data, like I had here in Zenoro, you have to put manually. Of course, it doesn't know where you put the data, but you can put the folder or whatever fair data archive you are using.
Okay, very good. And leading on from this, can IceBear be applied beyond crystallographic pipelines and used basically as an electronic lab book?
Yeah, we have discussed this. I don't know how feasible it is, because it's basically it's not to replace an ELN. My team is actually running ELN on the side, and we can put links to that ELN where you have crystallization or purification experiments or whatever. So I wouldn't maybe replace ELN, but if you have data management where you have a sample, you do something with the sample, you reach the data is somewhere, then tracking that process, what did you actually do to the sample and so forth, there it's useful. So we have discussed with tracking cryo-EM samples, SACS, which is quite natural because it's in IcePyB, so there's a connection also there. But also some biophysics could be doable, but we haven't really implemented such things yet.
And then going back to PARPs, so William has a question. If I remember your 2018 paper correctly, all the mutations that disrupt bridging of 2 DNAs also disrupt PAR formation. Isn't this in favor of the interpretation that double-stranded breaks are a good activator and that bridging of DNA ends by PARP2 is physiologically important? Could you elaborate, please?
Yeah, I think so too, but there is a we have also shown that the stoichiometry seems to matter to some extent, which now when we have these structures available, then we had the SACS papers previously about this. So it also depends a bit when you have we know that likely this happens, but it doesn't matter if it's a nick. Nick is very robustly activating the enzyme. And this is what we often use. We use a nick as a model. But when you want to prepare a complex for structural studies, then this blunt end DNA works the best because the hairpin seems to have also some affinity and so on. And if you mutate this VGR domain, then you affect immediately the affinity to the DNA in general. So dissecting that away from the activation is not super straightforward, maybe always, because we have to use the same stoichiometry, so we cannot boost the concentrations.
Okay, so thanks very much, Lari, for all of your answers and explanations. Now it's time to turn over to the last speaker of today, which is Yuhar Rovenan from Joensuu from the University of Eastern Finland. Take it away, Juha.
Okay, so I'm Yuhar Rovenan, and I'm a manager of this mass spectrometry site here. And Professor Janne Jänis is a main contact person if you have any scientific or technical inquiries afterwards. So we are located, as our university says, in Eastern Finland. And I will start by showing our major instruments. So we have 3 actually. And this is the first one, ultra high resolution mass spectrometer. And so it has a very large magnet, 12 Tesla. So it's very powerful for isolation of different signals. and you can use it for analysis for very complicated mixtures, for example. There's so many additional smaller instruments, like we have the LC and the microchip basic ionization robot, which can be used, for example, ligand-based screening for drug design. And also we had so-called desorption electron spray ionosource, which can be used for ionizing from samples from solid surfaces and so on. This can be used also for analysis of proteins, but there's an upper limit, something 200 to 300 kilodaltons. And for example, antibodies can be measured, but not larger complexes. They are much more difficult. The second instrument is the ion mobility mass spectrometer. It doesn't have as high resolution as the previous instrument, but this is one benefit. It is that these ions are separate based on their size and shape, which means that if you have, for example, small molecules and isomers, you can separate them. Or if you have proteins and they have very different conformations, major conformations, you can also get signal from them. So it gives additional information what this previous instrument doesn't give. And the third instrument, what we don't have yet, but we are going to have it soon, is ultra high mass range mass spectrometer. And this is especially useful for measuring very large proteins, even viruses up to 20 megadaltons. And this is a very new instrument, and the benefit is based on the good desolvatation. It helps when you are measuring very large ions. I will give one example of our test measurements soon. So this slide summarizes major applications. As I said, we have this high resolution mass spectrometry, and this can be used for intact protein characterization. For example, does your protein correspond to your sequence? And if you have any post-translation modifications, you can get information from them. In some cases, you can use also it for de novo sequences. We have sometimes determined primary structure from protein material. It's also possible. But as I'm a protein crystallographer, and I consider this native mass spectrometry the most interesting additional method to offer unique information about interactions. For example, protein ligand, protein metal ion interactions. And also it's possible to measure these valuable KD values, these equilibrium constants for these interactions. And as I said, we can use also the ligand screening by using small molecule compound libraries. And this ion mobility mass spectrometry offers information about the conformations for proteins, for example. And you can also calculate so-called collision cross-section from your protein structure. And then you can compare this calculated collision cross-section from your measurements, and then find correlation between your structural studies and this information you will get from ion mobility mass spectrometry data. I will show by using different examples what does this mean in practice. First is what does this ultra high resolution means. So here we have the example of small molecules, enzymatic reaction. Here we have a substrate, deoxyribose. It's cleaved to two aldehydes by this DERA enzyme. Here we have an acetaldehyde, which is very volatile compounds. So that's why we have made the derivative from this, all these three compounds. And here you have derivatives. And these are the peaks. And high resolution means that you will have a mass accuracy in the millidaltons. So you can use this high accuracy mass to calculate direct elemental composition of this peak. That's why you can identify that this corresponds to this one and so on. When I was preparing this slide, I recognized that this is this peak. And what is that? So I took this mass and made a database search, and it reveals it's from TRIS buffer. So this shows how useful this high-resolution mass spectrometry can be in analyzing small compounds, even from complicated mixtures. So you don't need any liquid chromatography separation. You will get all this information from one single mass spectra. And then what does it mean in the protein world? So here we have so-called deconvulated mass spectrum. So you have only simplified mass spectrum. So you have only one charge state here. And this blue one is the magnification from this peak. And as you can see, the small peaks, which the difference is 1 Dalton or proton neutron. So this is called isotopic resolution. You can calculate this mass spectrum from your protein sequence. As you can see, there's 571 carbon atoms and so on. So that your sequence corresponds to your measured mass. Here you can see the small modification, and terminal glutamine has cyclicized. So it's a minus 17 Dalton difference. And there's a minor component of that. So if you have any modification, you can see it from this high resolution mass spectrum. For example, if your disulfide bridges are formed or not, you can see it in the mass difference. Then, if you are going to folding of the protein, you can use native mass spectrometry to probe state of your protein. Is it unfolded or folded? Normally, proteins are measured in denaturated state in organic solvent. You have very high charge states. This x-axis is m over c, so that's why you have in the left part. But if you are making measured your mass spectrum in water, in ammonium acetate buffer, you will have a so-called soft ionization. So it preserves your native folded protein. Here you have the monomeric enzyme, here you have tetrameric. So tetrameric state is preserved. You will have even a small fraction of octameric protein. Also, this protein has some magnesium ions, which are still in these peaks. So this is a difference between the unfolded or denaturated protein and folded. Very clear difference in the mass spectrum. So this shows the mixture of different states in mass spectrum. So this is this DERA enzyme. And you can see the single spectrum folded monomer. So it has this kind of Gaussian type of distribution. Then you have a dimer, even trimer. And then you have also unfolded fractions here. So this shows the strength of this native mass spectrometer, so it can then reveal different states into some same single spectrum. So you will get a clear signal for all of this. And you can use this single spectrum even to have a rough estimate of the KD for dimerization. It's in this case a little bit of 1 micromolar. Then we have these complexes, protein-protein complexes. So this is the bovine milk allergen BOS-D5, which is also called as a beta-lactoglobulin. There's a crystal structure, it is here. And the monomer-monomer interface is not very large. It means that in solution, it dissociates monomers easily. So this is the simulated amount of dimer, which increases when your protein concentration increases and amount of monomer goes down. There will be equilibrium constant. We have made a titration by using native mass spectrometry, and it is calculated to 20 micromolar. And this is the typical mass spectrum from the protein which dissociates easily to monomer. So it exists as a mixture of monomer and dimer in solution. Red peaks are from dimer and blue peaks from monomer. Sometimes it's quite difficult to measure this kind of weak complexes, but native mass spectrometry has such a large dynamic area that you can do it. If you are measuring the stronger dimers, like this is allergen from horse dander, it is a much more extensive monomer-monomer interface, and this KD is in the nanomolar range. And also these curves are shifted to low concentration. But even here, in the 40 micromolar concentration, you can get signal from monomer. It means that this even this dimer follows the law of max action. So it's a small dissociated amount monomer still left, which can be observed by using a high resolution instrument. And this is now measured by using this new instrument for testing. And this large protein is coming from the laboratory of Minna. It has a molecular weight of 255,000 Daltons. And this tetrameric protein has 11,200 carbon atoms and so on. It has an iron sulfur cluster. And now this calculated mass corresponds to observed mass in the error limits. And this shows the quality of this very large protein. So you have very good signal noise relationship here. So we expect that by using this new instrument, you can get very useful information from very large protein structures, even from viruses. So this extends the possibility to utilize native mass spectrometry for even large protein complexes. Then we have protein ligand complexes. So here we have an antibody FAB fragment, which is specific for thyroid hormones. And here we have low affinity ligand. We have here iodine atoms. And here we have the simulated curve. And we have made again this titration and calculated this KD value 0.26 micromolar. So this shows the behavior in the mass spectrum, that there's a one charge stage of the apoprotein which goes down when the ligand concentration is then increasing, and then you'll have the complex signals. So you can distinguish this protein without the ligand and with the ligand and get this kind of nice curve to get very accurate KD value for this ligand binding. Then we have this ion mobility mass spectrometry. So this is an example of a small zinc finger. And this is the normal mass spectrum here. And here have the ion mobility gram. And now you can see that the two peaks are separable. And you can calculate the so-called collision cross-section from your protein structure. And in this blue one is from this measured value, and this red value is from the protein structure determined by using NMR. So these values don't correspond very much each other, but in NMR you have only one single structure. But this shows that you have actually two different stereoisomers, which are probably due to that this coordinating cysteines and histidines are somehow, there are two different ways to coordinate there. And this NMR structure represents only one of them. So this ion mobility is proportional for the collision cross-section, which can be calculated from the three-dimensional structure. And then you can find correlation between your protein structure and this value what we will have here. And if you have a major conformations, you can get the relative proportion from this native ion mobility mass spectrum. And another example is again from this beta-lactoglobulin. Here you have this mass spectrum. Here you can see the one additional peaks which is due to the covalent modification in the protein. And then here you have this ion mobility gram. This blue one is for your monomer and red one from the dimer, and you can calculate again this collision cross-section from the mobility. And then try to find the correlation between your protein structure and this mass spectrum data. Yes, this was my presentation. Thank you.
Okay, thanks very much indeed, Juha. So Isabel has a question. She wants to know more about protein DNA complexes.
Yes, certainly. There's no reason why it would not work.
Okay, and then Maria Anastasina would also like to ask a question.
Yes, hello. I wonder, would it be, well, I'm interested in detecting a small molecule binding to a virus, provided that not necessarily all the binding sites are occupied with the molecule. Is it possible to do it?
To probe small molecular binding to viruses. Actually, when we made these test measurements, Hanna Oksanen from Helsinki provided us viral particle, and there was indeed small molecular bound to this viral proteins. And molecular weight was some 200, 300 Daltons. We suspected it is some lipid probably. So it is probably, to answer for your question is yes.
Okay, sounds good. Thank you.
We had one interesting metalloenzyme. We have observed that this metal, which was identified before to be involved in the catalysis, is not the right one. So our mass spectrometer measurement revealed this totally new kind of metal. So we had just submitted the paper that I was not able to tell what enzyme is and what metal is that. But in normal protein crystallography, we are not necessarily sure what your metal is bound to the protein. And it was completely different, which was assumed before.
That's
and also in that case, we were able to determine the KD value. This is not very common in metal enzymes to get this KD value for metal binding.
So now we've got lots of questions coming, which is great. So I'm next going to take Arno, has put his hand up.
Yeah, so what are the tolerance of the new instruments towards sample composition? I mean, in terms of salt, I mean,
Salt, okay, if you have salt. Salt, so this is important questions. Sodium is a bad thing for native mass spectrometer measurements because it binds to your protein and very, very easily. And that's you will get the very broad peaks because it loves to bind sodium. And because it's so high accuracy, you will get a huge amount of sodium atoms in your protein and very broad signals. So we have to dissolve it in practice. But then there's the question that if your protein is stable without the salt or sodium or other salt, and it's depending on concentration, as I said, so you will need to have a high concentration so that your protein is assembled if it's following the law of mass action.
But my question was regarding the new instruments. I mean, are they more tolerant than, I mean, are there improvements? I mean, new instrument, high concentration.
You are not able to have a salt even in the new instrument because you will get the signal from this salt bound to your protein.
Okay.
So you have to remove salt because then it's bound to your protein and you will get the very broad peaks.
Yeah. Okay. So let's move on. Thanks very much. So in the chat we have a question from Bruno who says, for the UHMR, what is the expected precision for complexes around 5 to 10 megadaltons?
According to specification, it should be possible to measure this kind of compounds, but what is the accuracy? That's more difficult to say because we have so little experience on that. But this protein matter provided by Hanna Oksanen, and we were able to find this small ligand there. It's lucrative that we could get interesting new information. I'm sure about that. So we need test and other, but it's a new opportunity to utilize this instrument for that kind of purposes, which has not been possible before.
Maybe this is something that Juha, you can add then to the Instruct-ERIC catalog description once you have an idea of what it is.
Yes.
Yeah? Okay, so let's continue. So Paula asks, in ligand binding studies, can you distinguish between irreversible binding and reversible binding with a very slow K off?
In principle, yes. If there's an irreversible binding, then the covalent link between your protein atom and your ligand. There's also you can see that also in the denaturated state. It's quite often that we have for example, we have done a biotinylation and then we will get the signals for different personality species. And of course, if you have a non-covalent binding and you make the denaturate this measurement, then you will lose your protein even if it has a very high affinity. So it depends on the K on a K off, but in principle the dynamic is quite large, so that I will be quite optimistic to get also information in that cases.
Mm-hmm. Okay, let's move on because we have lots more questions. So how do you know that the monomer is unfolded to folded protein by MS?
How do we know that protein is folded or unfolded in mass spectrum? So in this red spectrum, it's unfolded, and this blue spectrum is folded. So it's a total different shape. And this spectrum, you have a folded, it's just a more narrow and high M/C values. In unfolded, you have here. So the shape of the denaturated unfolded species and folded species is very different. And here you can see the both in the same spectrum.
Exactly. And then there's a couple of people asking about membrane proteins by mass spectrometry. Are there specific requirements? What about differentiating between different detergents?
There are. We have not done that, but of course, but this Carol Robinson, she's in Oxford now. She's specialized in this area. Yeah. And also, I think also some other groups. Yes.
Yeah, and I think Carol's also offers facilities through the Instruct-ERIC as well. So it's worthwhile contacting her. Then we have Atushi is asking, you haven't mentioned it in your talk, what about mass spec-based footprinting using hydroxyl radicals? Have you ever carried out the method? I've heard a lot of special techniques are necessary. I guess it's hydrogen deuterium exchange is talking about.
I had that in my original slide, but I removed that because I consider this quite challenging technique, and directly speaking, the information what we will get from quite extensive experiments is very limited corresponding information you will get from other structural studies such as protein crystal for electron microscopy. So I would prefer these methods for characterizing protein-protein interactions rather than the hydrogen deuterium exchange. But this is my own opinion.
Yeah, you're allowed to have opinions. So Noel asks, which instrument could we use for analysis of methanolic microbial extracts for small molecule secondary metabolites?
This FTICR.
FTICR, there you go.
Yes.
That was easy.
I can mention that we have measured, for example, this bio sample which contains thousands of compounds. So we are able to distinguish one spectrum, thousands of different compounds. So it's such a high resolution without any chromatographic separation.
Fantastic. Okay, I'm going to now close the session as we're going way over time. Thank you all very much indeed for your active participation. Thanks once again to all the speakers. Great job, guys. And we'll see you all again in another month's time in Instruct Centre France, part 1.